So today we have Dr. Danny Glavin to talk with us. He's an astrobiologist, and where is Danny? He disappeared. There he is. All right. He's an astrobiologist and senior scientist for sample return in the Solar System Exploration Division at Goddard. He looks for he researches the chemical building blocks of life in extraterrestrial materials, including meteorites from Mars and samples returned from the Moon and asteroids. He's currently the project scientist for the OSIRIS-REx Mission, which collected samples from the asteroid Bennu. He's a member of the Surface Analysis of Mars, SAM, instrument science team on the Curiosity rover that has been exploring the Gale crater on Mars since 2012. He's very busy, Danny. He's also a science member of the Jackson Martian Moons Exploration Mission, which is going to be launched this year. And that's going to hopefully return samples collected from Phobos, one of the moons of Mars. So in recognition of all his work, Danny has an asteroid named after him. It's asteroid 24480 Glavin. There you go. He's received a variety of awards: Antarctica Service Medal, the Meteorological, I have a hard time saying that, Society's Near Prize in 2010. He was Goddard's Innovator of the Year in 2007. Yes, very nice. And he was elected a Fellow of the Meteorological Society, a little better. And his research discoveries have been featured on National Public Radio, PBS, NOVA, Discovery Channel, and in several books. So Danny, welcome to GRAA.
Thank you, Carl. I think I just had a bout of imposter syndrome there when we were reading that list of things. But it's true, I'm really busy. Actually, when Carl asked me to give this talk, was it back in August or, I don't know, spring time frame, I said, if I'm still here. I mean, it's been quite a year. But I'm here today to talk about NASA's first asteroid sampling mission, asteroid sample return mission, OSIRIS-REx, which has been quite an amazing ride. OSIRIS-REx, like most NASA missions, is an acronym. Origins, which I'm going to talk a lot about today, origin of the solar system, origin of life itself, potentially what we can learn. But there are a lot of other science objectives of this mission: spectral interpretation, providing ground truth for all the spectroscopy measurements being made on the asteroid population, resource identification, mapping the surface for water and other hydrated minerals. Security, this is actually a big one. NASA has an entire planetary defense program to track these things. And it turns out Bennu is categorized as a potentially hazardous asteroid. It has an Earth-crossing orbit with a non-zero chance of hitting Earth in 2182. None of us, of course, will be around then, but maybe our great-great-grandchildren will. So something to pay attention to. And I will say, everything we've learned, that probability is not down. So that's good news. Next slide here. I'm a big Orion fan. It's always been my favorite constellation. This image from JWST is just incredible. I mean, we're actually seeing the formation of a solar system, right? You've got a disk of material, there's the orbit of Neptune for scale. And understanding, we look to space, understand how our own solar system formed, but we can also study asteroids as well. These are the remnants of solar system formation, so we can learn a lot about the history of our own solar system from these samples. Another hypothesis is original life, that in fact, life wasn't homegrown. It didn't just sort of sprout from organics made on the Earth. These building blocks were in fact delivered by asteroids and comets, giving rise eventually to life. And so we want to test that hypothesis as well by studying asteroid Bennu samples. And this is something that I remind folks of. We can't really find we can't understand the chemistry that led to life on Earth by studying Earth rocks. We think life started on Earth around 4 billion, maybe 3.8 billion years ago. And the rocks that are of that age are just completely destroyed. They've been altered by plate tectonics, they've been weathered. Of course, there's biology everywhere on Earth, so they've likely been contaminated with life itself. So you can't really look at a rock and say, okay, now I understand how life started. There's the prebiotic chemistry. But with asteroids, you can't. These are 4.5 billion-year-old samples, right, that provide a unique record of the prebiotic chemistry in the early solar system. And more importantly, they haven't been contaminated because we've gone out there, we've collected them and kept them clean. Meteorites, when they fall, they get heated by atmospheric entry, they hit the Earth, and they're immediately contaminated by bacteria and all this stuff. So it's really important that we do missions like this. Okay, so I just have to show this animation. So we're actually at Asteroid Bennu. On the left is a reconstruction animation of this touch and go sampling process. So we had this long arm with a tag SAM head that looks like a 57 Chevy air filter. We fired nitrogen, compressed gas to stir up the regolith. You can see we made quite a mess there. On the right is the actual images, right, stacked images of that touch and go. And this was fully autonomous. I remember we were sitting in a room at Goddard. This was during COVID, by the way, so we're all like 5 or 6 seats apart, masks, and kind of looking to see who's coughing and sneezing and all that. But at that time, our attention was on this, and we were just waiting for the data to show that yes, we had collected sample, and indeed we have. I was talking to Bob Jenkins over there, who was involved with this, and there were just so many small miracles that had to happen for this mission to be a success. All the way to the end, when we brought the samples back and they landed in Utah, we didn't know if the parachute was going to open. There was an anomaly there. So, and as Bob said, some of this is just takes some luck too. You can plan and test the heck out of everything, but you need some luck. And we got a bounty sample. We were very excited. Over 121 grams of material. May not sound like a lot. We would fill up a small cup. But for the kinds of analyses that we have planned and looking for the building blocks of life, this is a huge amount of material. And more importantly, it's been kept clean. Right? It hasn't been infected by biology and other organics. So there you see the tag SAM head open. I don't know if you recall, but there were a couple of stubborn screws that had cold welded in space that had to design a special instrument to open it that met the contamination requirements of the nitrogen cabinet where these samples were kept in. And so that took some time, but we finally got it open. And there are actually different rock types as well. So this is the hummocky stones. These look like cauliflower. They're kind of friable. And then there's some more angular shaped stones. All of this is very dark, very, very black stuff. With the exception of this model lithology, which contained this white kind of coating. And that was actually a big surprise. That was something we didn't detect from orbit. These other two, we kind of matched to the different boulder types on Bennu, but this model was different. And it turns out it's a magnesium phosphate kind of crust. And phosphorus is really important for life. Kind of a rare mineral. We don't find that mineral a lot in meteorites, so that was really an exciting find. So we had these different rock types, abundant carbon, nitrogen, and clays. 80% of this material is alosilicate clays, so water rich. And so this sample is really, for me as an astrobiologist, it's really an astrobiologist's dream. I mean, this had all of the right stuff, so we picked the right asteroid for sure. These samples have been distributed and are still being distributed around the world. Over 200 scientists, and I forget how many laboratories, a lot of different laboratories. I'm going to talk about some of the stuff we've done at Goddard, because we're near Goddard here. The sample's been analyzed with over 65 different analytical techniques, and of course, this number is going to continue to go up. This is the benefit of sample return. These samples will be around for generations using techniques that haven't even been invented yet. I give folks the example, I wasn't even born when Apollo happened, and we recently analyzed an Apollo 17 core that had been kept sealed for 50 years. And we detected amino acids in these soils that had never been discovered before because the technology didn't exist. So again, this is really the power of sample return. Even though OSIRIS-REx is coming to an end officially at the end of this year, for me, it's never going to end. And in fact, more than 70% of the sample will be saved for future generations. And just to hammer the point home about why sample return is so important, we're using synchrotron radiation facilities right now to study the organic composition of these materials. You're never going to fly a facility into space, right? So you need to bring samples back. This is just kind of a cool picture. So here I am, all masked up. This is we call it the white room. It's not officially a clean room because we don't get it certified, but it's a clean room. We actually got a 52 milligram sample. You see it here. Over here. I hope I'm not blocking. This dark material inside these, actually use these glass slides like you'd use for a microscope. And it turned out that was a really good, clean way to ship these things. And then that slide was inside this Eagle stainless steel container, which was sealed under high purity nitrogen. So again, these samples were never exposed to earth air. Really important. And then we, of course, opened it up and quickly analyzed them, and our goal was to search for these chemical building blocks. You probably remember this. There was actually a press conference. I was at Johnson Space Center called the Sample Reveal. I can't remember when it was. It was, I guess, late 23, maybe early 24. Been a bit of a blur. But the exciting kind of findings here was this organic carbon. You see these little glebs of like light. These fluoresce under UV. So it's kind of like the crime scene investigator, right? With the black light, looking for fluorescent organic stuff. Not getting into too much detail, but this thing's loaded. And Simon Clement at NASA Johnson Space Center said he's never seen anything with this much organic carbon in it. And you see this plot. This is nitrogen and carbon. You see Bennu here, almost 5% carbon, 0.25% nitrogen. It really stands out among all the astromaterials we've analyzed. So truly a special sample. So we started digging into this, and we were initially interested in looking for amino acids. These are the building blocks of proteins in water extract. I see a hand coming up.
Just a quick question on the other astromaterials. Are you talking about moon samples, or are you talking about other asteroids? Or?
Yeah, moon is way low. There's really not much carbon. These are mostly carbon-rich meteorites. So among the most carbon-rich meteorites, Bennu still stands out.
Is it because we got it off the surface, the meteorites, or maybe the surface burns off?
That's a good question. I might address that a little later. But yeah, it turns out we don't have a lot of meteorites in our collection that are Bennu-like, and we think it's because it's just so friable. Most of the stuff probably didn't survive atmospheric entry. It's a good question. So again, we were looking for amino acids, but actually the big surprise was ammonia. We were not expecting that. So here you see in the blue bar here, we found ammonia, and we basically say we were making Bennu tea. So we basically heat these samples up in water and then analyze the liquid extract. And ammonia, when I first saw this, I'm like, oh my God, the hydrazine thrusters, and Bob probably knows this, one of the major byproducts of hydrazine is ammonia. I'm like, crap, we basically contaminated the sample with ammonia. But we had the foresight before launch to measure the nitrogen isotopic composition, the 15N, heavy nitrogen over 14N. And we knew what the hydrazine was, even before launch. And we measured Bennu, and it was like a plus 180 per mil, so a lot more of the heavy form of nitrogen than what we found in hydrogen. So right away we could rule out contamination. I'm like, okay, let's submit this thing to Nature Astronomy. That's the data point I needed. We think ammonia may be coming from salts, maybe associated with clays or other organic matter, maybe these magnesium phosphate, the white stuff as well. They could be a mineral fluid inclusions. The nitrogen 15, in addition to showing that it wasn't terrestrial from the thrusters, it also points to an outer solar system origin. You tend to have more of the heavier form of nitrogen in the outer solar system. I'm talking about beyond the orbit of Saturn, right? Way past the water snow line. So that's pretty cool. And then ammonia is an important precursor for the formation of the amino acids and nucleobases, which I'm going to talk about. Angel Moharo, who's an NPP in our lab, really a superstar, developed this new technique. It's a pyrolysis gas chromatography mass spectrometry technique, actually inspired by some of the SAM work and the wet chemistry that we're doing on Mars. optimize this instrument so you only need a milligram of sample. You can barely see that with your eye. And from that milligram, he heated it up and was able to detect all 5 nucleobases found in our DNA and RNA, so the entire genetic code, pretty incredible, as well as 15 of the 20 protein amino acids that are common in biology. In a single milligram. I mean, again, the power of the sample return, right? Now, we're still looking for these compounds on Mars, and you know, it's hard. It's hard to make these measurements in situ. The timing of this paper, which came out in PNAS, couldn't have been better. So it was published 2 days before Thanksgiving. And tryptophan, as we all know, with turkey, it was too hard for CNN to pass up. So they got word of this. They said, oh my God, there's, you know, Bennu's a big turkey or whatever. It's kind of the tagline. Like, well, you know, but tryptophan itself is really interesting from a prebiotic chemical perspective. This is actually one of the more complicated protein amino acids. We still actually don't know how it formed in the Bennu samples, but it's got this aromatic ring. It's complex. But what it's saying is that, you know, the Bennu parent body was a chemical factory churning out, you know, even some of the most important chemical building blocks of life. I remember Angel was so happy because he said, you know, not many people had read the PNAS paper. After the CNN article came out, he said there were like thousands of views of this PNAS paper, so he was really excited. Another really important finding, so sugar. So I talked about amino acids for proteins. DNA and RNA have these nucleobases, but sugar is actually an important prebiotic organic chemical for life as well. Yoshi Furukawa at Tohoku University was partnering with us. Actually, there's a very good collaboration between NASA and JAXA because we're sharing samples. They have another mission called Hayabusa 2 that went to a different asteroid called Ryugu, and NASA and JAXA have this high-level agreement to share a certain percentage of the samples, but also the scientists. And so we started working together, which has been really fruitful. And this is just one example. But Yoshi was able to measure ribose. So ribose is a 5-carbon sugar, you see it in gray here, which is a fundamental component of ribonucleic acid. Some origin of life scientists think that RNA was actually the first genetic material on the early Earth, before cells, before DNA, before protein, because it can self-replicate and all those things. So the detection of ribose was huge because, you know, we had the phosphate, we knew about the nucleobases, but the ribose was the missing component. So now we have all pieces of RNA in Bennu, which is incredible. Now, how those came together, that's a story for another day. But at least we have, you know, we have the puzzle pieces. We have the grocery list, if you will, for life. Another exciting finding here is glucose. This is the energy source for life, right? Involved in metabolism. It can also link together to form chains, plants, cellulose as well. So another important sugar in life. And this had never been found in any meteorite or astromaterial before. So this is the first time that we've discovered this. Perhaps, you know, because bacteria gets in there, maybe it just ate all the glucose in meteorites. We don't know. But this, we know it's formed in space. Okay, I want to turn a little bit to mineralogy as well, because it's important to understand the context of the Bennu parent body and what was going on in the early solar system. And this was a paper that came out in Nature. Actually got the cover, but they found these evaporite salts. This is false color, they're not really purple. But these evaporite salts are actually, some of them are called trona. Trona is a small town in the Mojave. And this mineral, which is a sodium carbonate, is basically, it's formed in evaporite lake beds. So in Mojave, there's one. But what was important here is that the sequence of precipitation. So by looking at these Bennu samples, you can see which salt mineral precipitated, which is it's incredible. So this was an evaporite brine fluid percolating through probably a 100-kilometer parent body in the outer solar system, probably past Saturn, that was maybe evaporating on the surface and depositing these salts. Ceres, you probably all know about Ceres. Some of these salts are very similar to what we see on Ceres. So we're starting to think that maybe Bennu was part of, maybe an ocean world, Ceres-like object originally. And again, we're learning that from, I can't emphasize this enough, like literally milligrams of material. Right? We're able to piece together this history, which is really exciting. Another thing I want to point out is the minerals themselves show that this aqueous alteration of fluid was at low temperature, probably around room temperature, so not too hot, right, to destroy organics. And you combine clays and carbonates and these salts and phosphates and these high pH fluids, and this is the ideal environment to make more complex chemicals. And we tried to illustrate that here. This probably should have gotten a cover, by the way, but it didn't. Jupiter beat us out for this issue of Nature. But we have the precursors. We have the ammonia, water, formaldehyde. We have the minerals, clays that can act as catalysts to produce organic compounds, nucleobases and amino acids. And so again, these chemical ingredients for life formed 4.5 billion years ago, prior to the origin of life on Earth, inside this giant parent asteroid. We've got 15 of the 20 protein amino acids highlighted yellow, and again, all 5 nucleotide bases. Okay, so I couldn't pass this up. This was another recent discovery. Anne Lynn at Johnson Space Center used a nanosim, it's called nanosecondary ion mass spectrometry. Again, one of these instruments that takes up half a laboratory. You're not going to fly one of these in space. But she was interested in supernova dust grains. And so it turns out Bennu has grains, silicon carbide grains, in fact, that have carbon isotopes, so a lot more of the carbon-13 than what we see in our own solar system, indicating an extrasolar, pre-solar origin. So these grains were probably formed in a supernova explosion, and then were swept in to our own solar system and the outskirts, incorporated into the Bennu parent asteroid, and now we're looking at them. So that's crazy. And so actually she found 6 times more of this supernova dust than we've seen in any other astromaterial. Carl Sagan had the saying that we are all star stuff. I can't think of a better example than this, right? We have a sample in our lab that, yes, there's a giant recycling program going out there. Okay, so just kind of putting this all together, we have all sorts of evidence now saying that, the Bennu parent body, not Bennu itself now, which is a near-Earth asteroid, but the parent body that Bennu came from, formed in the outer solar system. Definitely past Saturn, probably Neptune, Uranus region, basically in the same place that we think comets were formed. And now it's in the inner solar system, right? It was once part of the main belt, it got disrupted, became this rubble pile asteroid, and then now it's in nearer space. And again, we're able to sort of piece together this history again from literally milligrams of sample. It's just, we would never be able to figure this out, at Bennu. So this is samples in the lab. There's a model called the Nice model, which I don't know, people who study solar system dynamics are probably more familiar with this, but the idea is that the giant planets were migrating, moving around, and they flung a bunch of stuff into the inner solar system as well as out. And Bennu was, one of those ocean worlds that got flung inward. So we're still arguing about this. Going into this, we kind of thought that asteroids like Bennu were probably fragments of comets, extinct comets that had lost all their orbitals. Kind of makes sense, carbon rich. But now it's looking like, well, maybe they're more, Ceres-like, maybe something more like an ocean world. We still haven't nailed this, but it's probably true that this object was just too big to ever become a comet. Right? Comets tend to be smaller. This object was probably much bigger. That being said, I think I really advocate for more sample return. We do need to go to a comet nucleus and return ice. Because we didn't have the ice, we lost all the ice in Bennu. There's a lot of information in there, so we need the comet sample return mission. I know there's some concepts that we're working on, but also Ceres, another good place to return sample. Let's really try to nail this. Yeah, I got a question. Stardust, yeah. And actually, I didn't show any data, but our group analyzed some of those dust grains, and we were the first to discover an amino acid, lysine, in some of those grains. The challenge with stardust is there was only a milligram total return, right, as opposed to 120 grams. And they flew through the tail at 6 kilometers per second. So those dust grains impacted the aerogel, and they devolatilized and were heated. And so the chemical record is you know, it's been messed with, let's say. So a combination of not a lot of mass, plus we don't have the ice and stardust. We really need to go back and get the nucleus material, in my opinion. Okay, so let's just kind of wrap this all up. I know I probably exceeded my 20 minutes, but you know, the fact that we're seeing the chemical building blocks of life in the Bennu samples, the fact that we know that this material was spread throughout the solar system is huge, right? This, when we were getting a lot of media attention, I was telling reporters, this increases the chances that we could have had another origin of life in our own solar system. Because this material was not only being delivered to Earth, it was being delivered to Mars, et cetera, et cetera. So I'm becoming much more optimistic that, you know, we'll probably find evidence for life, you know, not ET, but evidence for the chemistry of life, even a bacterial cell, even in our own solar system. Bennu's parent could qualify as a habitable world, right? It had this cool mild temperatures, salty environment, alkaline pHs. We had the wet, the clays, all the elemental building blocks, carbon, nitrogen, phosphorus, you know, chomps, we often say, and then even more complex organic molecules essential for life. So here's the million-dollar question, right? Why don't we see even more complex molecules? We had the right environment, we had the building blocks. Why don't we see DNA or RNA, right, or peptides or proteins? And I'll just leave this with you guys. What conditions did the early Earth have that the Bennu parent body didn't, right? We know life started here. And so I think if you can answer that question, you're probably going to get the Nobel Prize in chemistry, right? Because you've basically solved the origin of life mystery. So I think this is going to keep me employed for a while. And I just want to send out a thanks to Jason Dworkin, a close colleague of mine. We both came to Goddard. He was a year before me, 2002, 2003. He recently left. You'll probably see him at one of these meetings in the future. But I kind of consider ourselves like you know, partners at a big company. I mean, we built the astrobiology lab together, so it's been tough seeing him leave. But he was instrumental with OSIRIS. He was a project scientist. He was leading the contamination control working group. You know, he did a good job because the samples are clean, right? We didn't contaminate, so he did his job, no question about it. And he also helped me out leading the sample organics analysis team. So I'm going to miss Jason, you know, seeing him on a daily basis, but we're keeping in touch by email and that kind of thing. I just want to thank him. And now I want to thank all of you for your attention. And again, this even though the OSIRIS is coming to an end at the end of this year, it's really not going to end. You're going to be hearing about end results for decades. I'm sure of that. And there's going to be much more to come. So thank you all.
The proteins are so complicated. How do they explain that they weren't broken down by solar particles and rays? You know, this is broken with fragments. And I always thought in the past, they said this stuff wouldn't survive in space with the radiation. Yeah, so that's a really good question. We've actually been doing some simulation experiments. We've been using gamma radiations as an ionizing radiation source to understand how quickly these components are destroyed. We've measured the cosmic ray exposure age of the Bennu samples that we're collecting near the surface. It's of the order of a few million years. So actually, based on our lab experiments, you don't actually destroy a lot of the amino acids and even peptides with that kind of dose. So that's the good news. But yeah, if you start getting into, you know, hundreds of millions of years where you're talking about mega rays, yeah, then you're going to start doing some serious destruction. But based on the cosmic ray exposure age that we've measured from the noble gases, we don't think that was an issue, at least for these Bennu samples. I don't know if that answers your question, but yeah. Yeah. Go ahead.
So at the end, you talked about not knowing how to get from the molecules you did find to more complicated molecules. Do we know the pathways to get to the molecules you did find?
Yes and no.
It's weird that
yes and no. Yeah, yes and no. So we haven't. I wouldn't say we put all the chemicals in a vial yet, you know, vortexed it and then we have RNA. We've So you can synthesize a lot of this stuff, right? So we're going to do that in the lab. It requires multiple steps and chemicals that probably weren't available on the early Earth and all this. You can cheat and get, say, one of the nucleotide components, and then you can amplify those pretty readily in this kind of environment. So that we've shown. But to answer your question, no, we don't know how to go from a mix of chemicals to something that can self-replicate. Not yet.
So I'll just say that I'm skeptical about your optimism that we will find on the second, evidence for a second dimension of our solar system. I tell you, but things like that are one of them, because although you found these molecules, you have no idea what's the probability that they would have been made.
Yeah. Well, one of the things I will say is that evaporative cycling, wet-dry cycling, we know from, like on clays, is really important for polymerization. So going from an amino acid to a dipeptide to a tripeptide, etc. It's not clear that that was really readily happening in the Bennu parent body, or not enough time. We think there's a few million years of aqueous alteration. Maybe needed more. That's one answer. This is why I like Mars, and I'm not going to lie. I'm frustrated that we've kind of sort of abandoned the Mars temperature. But we're in a lake bed, right, where you probably had this wet-dry cycling, not for a few million years, but tens of millions of years. You had the ingredients. Maybe we can learn something more about these other steps that we're not able to reproduce in the lab. And we have a natural example of that right now. Yeah.
Yeah. So, I'm trying to figure out what comes first, the OSIRIS-REx project, or then the selection of that particular asteroid?
Oh, maybe that's a Bob question. He came in early days.
It was the scientist, the original principal investigator that chose that as a target, and then proposed it with the mission.
And what evidence, what lunar evidence they had that said this was a good candidate?
They were just really looking for very carbonaceous asteroids, and they felt like Bennu, which when we were writing the proposal, Bennu wasn't its name. It was RQ 1999, RQ 36. They felt like out of all the candidates, and there's a really interesting paper that was put together. You know, imagine you start with all the asteroids that are out there. It might be scientifically interesting, but then ultimately you start to lay in a lot of other constraints, like what can you get to with a reasonable amount of propulsion and onboard capability and fit within the budget. So anyway, Bennu was what popped out.
The only one, their backup?
There was one other backup.
There was an earlier proposal for OSIRIS.
That's right, discovery proposal.
Was that also going to Bennu?
I think so, yeah.
It was.
Yeah, I think I'm remembering that pyramid where you have, over 1 million asteroids, and then you had to have the right orbit, so we weren't going to go out to the main belt. It had to be an Earth asteroid. It had to be large enough so that it was spinning. So the small ones tend to spin, and there's no way, try to jump on a merry-go-round really fast, right? That wasn't going to happen. And then, as Bob pointed out, in the end is it carbon rich? And I think there were only 5 or something that were even viable. So you went from 1 million to 5 pretty quickly. Yeah. Yeah. Yeah.
Can you say anything about OSIRIS-REx? What's happening with it? Anything? And just tell folks about office.
Yeah, so I'm not on the Apex team, but after we dropped off the sample return capsule after OREX in Utah, it flew off, and it's going around the sun a few times, and it's waiting for Apophis. I think Apophis is like a 340-meter asteroid, smaller than Bennu, but it's also potentially hazardous asteroid. In fact, it's going to come. It's April 13th. Is it a Friday too? There you go. Actually, 13 is a lucky number for me. My wife's birthday is 13. But yeah, 2027, 2029. Okay, thank you. Getting my dates. Yeah, you're right. And so the OREX spacecraft is actually going to rendezvous with Apophis just after it passes Earth. And I think if you're in Europe and Africa, you're going to see this with your naked eye. It's like, closer than some of the high altitude satellites. I mean, it's closer to geosync. Thank you. So you'll be able to see it with the naked eye. And we're actually going to go down. We don't have a TACSAM collector anymore. We don't have one, so no return. but it's going to go down and use the thrusters to excavate the subsurface. We still have the instruments, the infrared OSIRIS, which was built at Goddard, infrared spectrometer. Andy Simon and Dennis Winter, you might know that. And so yeah, we're going to get some more chemical information even from the asteroid. We're going to see how the Earth perturbed. You know, there's this thing about like asteroid quakes. This is getting outside of my expertise, but we're going to probably see evidence for boulders moving because of the interaction with Earth's gravity. And I mean, for planetary defense, it doesn't get much better than this, right? So we need to understand these things. It's not it's not a carbon-rich asteroid. It's we call it S-type, ordinary Conrad. But it turns out those are some of the more populous ones. So we need to understand this type of asteroid as well. And thankfully, it's back in the, I think, the budget. It was canceled in the PR, I think. Yeah, but you don't turn a spacecraft off. Anyway. It survived, so the instruments, it actually got way hotter because it had to get closer to the sun. And it got way hotter than we tested some, but I'm told everything's fine. It survived the bake out, so it's cleaner than yeah. Anyway, no, that's going to be very exciting. I think I might have to go to Europe to check this out. Yeah.
You said 2029?
2029, April 13th.
For a while there, I thought they had 2026 as a goal.
I don't think so. 2029, no. Chris is right. Chris is right. I don't know why I had 2027. Too many dates.
Pick up on a question that was raised earlier about following the path of your discovered molecules towards something that would be verifiable in pre-life form. Have you ever connected any of these results with work done by Carl Sagan at Cornell 30 years or so ago? Reconstructed these so-called tolins from synthesized molecules of precursor to life.
Yeah, so, yeah, Carl Sagan, that's how I got into this area. I mean, it was just, you know, the cosmos and so inspirational. Actually, I did my PhD on he had another hypothesis about the exogenous delivery, and there was a couple, Chivin Sagan was a paper that I always reference, where he's looking at these interplanetary dust particles that could have delivered the organics. And so I actually based my thesis off understanding if amino acids could survive that. That's a whole other story. The tholins, I mean, dragonfly, right? I mean, type. These tholins are probably produced in abundance there. And so that's going to be exciting. I think dragonfly lands in 2034, will arrive, launch 2020. That might be the 2027, launch in 2027, and then, and so I think we're going to learn a lot more about Bowman type chemistry. I don't know if I answered your question. I think there's still a lot we don't know about how to.
His work at Goddard by measuring the infrared spectra of those tholins.
Okay. All right. I'm sure some of that data will be brought back to Revival when we're looking at dragonfly data on type.
The data that came back from Cassini mission.
Okay. Yeah, Cassini was another amazing, incredible opportunity there. Yeah, I wish I had the answers for you about how this happened. But what I can say is that before Bennu, we were skeptical about the data we were getting from meteorites because we know bacteria get in these things and they grow and they alter the signals and the chemistry. And so we weren't really sure what was formed in space versus what was terrestrial contaminant. So this is a big leap forward. We can trust the results and tie it back to the existing meteorite data and say, yeah, that was right, or no, that was wrong. And so yeah, we're pretty excited. And again, we're just getting started. So we're able to tell you what we know based on the analytical capability today, right? 30 years from now, I mean, maybe there are peptides in there. Maybe there are tiny pieces of nucleic acids, but we're not sensitive enough to identify those. So stay tuned. Maybe there is, I think Bennu has more to tell us. We're not ready yet. Yeah, thank you.